So you want to keep that handout, and you will be able to use it on the quiz. I might ask you to sort of recreate this reasoning, maybe extend it a little bit on the quiz if you choose. But the other thing I want to talk about is as a biochemist, so this is one way as a biochemist looking at the chemistry of antibody binding, okay? What else can we do when we look at the chemistry of antibody binding? And I do want to say, antibody maturation, we talked about how it's a lot like what the llama and Wells did, and that's a lot like evolution. And so what are the differences and comparisons we can make? Here is a paper that actually talks about antibody maturation in terms that I would accept for evolution of predictability. So realize when I say evolution of predictability, I am using a broad version of evolution. I want to find interesting things. And so nobody's ever done this paper. I think as immunology can be a bit daunting. But if you are looking for presentation 4 and you're interested in getting into antibody evolution, if technically affinity maturation, I would accept this. I would totally accept this for the final presentation. So, but that's a ways away. The thing is, this is about predictability because it's about being guided by the intrinsic mutability. How evolvable or mutatable is something intrinsically? That's a question of predictability. And so realize that if you might be useful to Google things like intrinsic immutability as well as and look for big systematic papers like this. Okay, so when you get down to it though, the topic of this last little bit is what is the structure of the antibodies that are able to form these specific interactions through these 6 hypervariable loops that come together. 3 from heavy chain, 3 from light chain. So what the loops can do is they can form many different shapes overall. You're not even looking at amino acid interactions yet. Like for a small little toxin type molecule or chromophore, they can form a little pit, and this is actually an example of each of them. It's hard even to see where the molecule is here. Here you can see they form a little groove for binding a peptide. And here you see they bind a flat surface. I think this one is lysozyme. Actually, I'm going to say they can bind small molecules. This is an HIV peptide. Of course, they could bind any peptide from 20 amino acids. And they could bind any protein made of 20 amino acids. You just have to form a surface that's complementary to it. So this is an example of a small molecule that an antibody binds, and we can zoom in on it. This is actually phosphorylcholine. Do you all remember what choline is? What chemical group should we be looking for if we have a choline? What functional group? You can see it up there. Or you can actually see, well actually what's easier to see, so you see right here, it's easier to see, you can see the choline and you can see the phospho. Can you identify the phospho? So with that in mind, once you see the phospho, look at the other part of the molecule. Do you remember what choline is? What unusual color do you see up here? You see the blue? And you see that the blue should be nitrogen, right? But it's bound to 4 things. That's weird. What's nitrogen bound to 4 things?
No.
Yeah.
Infinable ring.
Yeah. Well, it's not necessarily in a ring, but it is unusual in that it has a charge. So, like right here, by the way, this is not part of the molecule. This is the tyrosine that's binding it. Once again, tyrosine. You see my friend, my favorite amino acid, and a tryptophan also helping out. We've got 2 tyrosines, so tyrosine's obviously better. But you have down here, you have the negative charge in the phosphorylcholine. The phosphate is here, and you have a complementary positive charge from the arginine. You also have, they showed them as hydrogen bonds, but sometimes people, are they really hydrogen bonds? I'm not sure. But you have some ring stacking up here. You don't have a particular negative residue unless this aspartate counts. This aspartate might be providing a negative that balances the positive over here. I would have to look at the distances to be more sure. But you want to take apart a structure like this. Look for the atoms, look for the charges, look for the bonds. In fact, H bonds. Now, H bonds are sometimes so they're so finicky. They really care about their direction. So sometimes you aren't sure if there's an H bond there or not, but they can really tie things together because when it's right with an H bond. It really forms a tight bond. And so you have the here's an antibody on the bottom binding an antigen, and all of these are H bonds. I like how he shows the H bonds as having a polarity. It has a red end and a blue end, positive end and a negative end. Because you can think of the H bonds that way. Remember that H is a positive and it's binding to something negative. So the other thing is when you look down on an antibody, sometimes the antibodies don't move at all. Sometimes they do this. This is the antibody crystal structure with no antigen bound. And believe it or not, you're looking down on the 6 loops. This is the conformation with antigen bound, but we hid the antigen. We just are showing the surface. But you can see where the antigen is. This is right here. This antibody, the loops have moved a lot to open up and accommodate the antigen. And there it is. Okay. So you can see that the loops can move, but they don't have to. And that's actually one of the big issues with predicting, even if you have a crystal structure of an antibody, you're not sure if it moves. Again, this is part of my postdoc research, another molecule, but we were asking the question of, we have this crystal structure, but it's frozen in place. How much does the molecule move, and is it important or not that the molecule moves? So antibodies can certainly move, and this is an example where if you just take the X-ray crystal structure, you don't see how it moves, but if you do molecular dynamics on it, you can calculate how it moves, and that can be very helpful to predicting what the antibody binds. So like here's 4 different antibodies that they looked at, and they have a little bit of the idea of the motion if you look carefully, but there's not that much motion. It's even hard to see on this. But I just want you to see there's a little bit of motion, but it's sort of small scale for these. And yet this small scale motion was really important to saying what shape is the antibody so that it can complement the antigen. Here's one, by the way, this one zooms over directly onto this. And they actually were able to calculate 73% of the time it's in the antigen binding conformation. 9% of the time it makes this shape, which will not bind antigen. And 18% of the time it makes that shape, which will not. So the flexibility can be important because it means you might think you have 100% of your antibody available. But it's moving around so much, you really only have 73% of it available for binding. Unless in the act of binding that the antigen actually forces it into the correct conformation. But that's another question as well. So you get into all sorts of questions when you start asking questions, how does the antibody move? And because these are too big for NMR, that's actually a whole research field, but I'm grateful for that because I like that I could publish papers on it. Okay, so this is just a couple of examples to wrap us up of antibodies moving and binding. Here's a chromophore. This is a yellow chromophore, so this would be like the tattoo chromophore. And if you had an antibody that bound a chromophore, they're able to study it because it changes color or something like that. They're able to use the color to study how it moves. And so you can see the 2 domains coming together. You can see the binding site right here. And if you look down at the top, you can see hydrogen bonds being made, even to bound waters. And you can see that pretty much it latches onto all of these external functional groups on the outside of the chromophore. Cool. But that leaves open the question of what's going on in the middle. Is it able to bind that part which cannot form any hydrogen bonds? And it turns out it can. Antibodies are very good at figuring out how to bind anything, find some way to form a bond. And they actually have a nice metaphor. They talk about it being like a palm and a thumb. Okay, so it's like you have the chromophore, and your thumb moves more than your palm, and your thumb can do this, right? So they say underneath the chromophore you have ring stacking with parallel rings of the tryptophan forming the palm that the molecule sits on. That's really cool, but then on the top you have a phenylalanine, and it does not stack in parallel, it stacks side to side. Still works because it's a hydrophobic interaction. But you see how it can kind of swing open and close. And it really affects how this chromophore binds. So they literally are talking about these residues. It's a complicated situation, but we boiled it down to just a few concepts, and that's the kind of concept you can use if you're a doctor talking to a patient, or if a biochemist trying to decide where to mutate and how. For example, I would be very curious to mutate that phenylalanine to a tyrosine and to mutate it to a tryptophan and see what happens. But that's just my thing. I see one of these things and I'm like, I want to mutate that. And I want to do it in a targeted way so I can see what happens. So that's pretty much, we're pretty close to the end. There's just a couple minutes more. I do want to connect this to the kind of antibody drugs you might hear about. And so the body makes a bunch of antibodies at once. And remember, they come from clones, but more than one clone is usually activated. So you usually have like a dozen clones at least being made. And your mixture, if you just take the antibodies out of a person, then they are polyclonal. They come from several sources. But if you're going to be a biochemist, you just want one of them. You don't want that heterogeneity. And so what you do is you isolate one antibody that comes from one B cell, and it is then a monoclonal antibody with a single sequence, a defined single set of amino acids. And then you can do all your protein chemistry stuff to it, because it is a defined protein. Monoclonal antibodies are useful as drugs, and of course you don't usually want to give polyclonal antibodies to people. There was one case early on in COVID where they were talking about using polyclonal antibodies, where they were just taking them from one person, not doing anything else with them, and putting them into another. Do you remember that? Does anyone remember what the name of that therapy was? Remember at least hearing about it? I'm blanking on what the name was, so that's why I'm not sort of asking, but I'm also asking, really. Antibody serum therapy, transferred serum. Serum was in the word. Because you took the serum, and again, they're proteins, and you take them from one person and put them into another. But it didn't work that great. You know, I think they were trying everything, but I think it might have worked moderately, but I'm not sure how much of a difference it really made. Because polyclonal antibodies are just worse defined systems than monoclonals. Whereas for all these different things, you have big trials, toxicity trials, and you basically, if you end up with MAB, it's a monoclonal antibody. So there's a code you can see for all these different drugs. And there's even you can tell which species it came from, which species it targets, and if you look up more, but this is actually in the name itself. They have these conventions for naming monoclonal antibodies. So we could say rituximab actually is a primate source, and let's look forward, it's X. Maybe it's a toxin. That doesn't, maybe it doesn't, oh, rituximab. Anyways, chimeric. It's chimeric. It's not just it's not a primate. So it's chimeric, and I think that tumor. It's anti-tumor and it's chimeric. You can actually play this game with these and try to figure out where they come from. The information is so transparent, it's even in the name itself. And so, of course, people were very, very interested. How do we make antibodies against the spike protein on SARS-CoV-2? And remember, SARS-CoV-2 is CoV-2. There was a SARS-CoV-1. I remember when that came out, I never knew that it would blow up so much for the second one. But I remember when the first SARS came out, I actually did a talk on it for Day of Common Learning. So that would be a blast from the past. But they are fairly similar. And in fact, there are antibodies that were made to SARS-CoV-1 that would bind to SARS-CoV-2. And in fact, the red are the only different residues, and the blue are similar. All the whites are identical between CoV-1 and CoV-2. So it's very similar. And in fact, you can color on the receptor binding protein, the RBD domain, that the antibody is binding. You can color these. The purple spheres down here are the ones that are different in the RBD. And the red is the antibody, and brown and gray are the spike proteins. So actually the differences are not even that close to the antibody. So it makes sense that an antibody that was developed to SARS-CoV-1 would be able to bind SARS-CoV-2 because not that different. There's some more information if you're interested in exactly what's going on with this, but I basically want to just tell you 2 quick stories and wrap up. When it comes to antibodies and SARS, my friend from church posted this, and she was like, "I can't. This is like you have to surprise me, whatever this is. This communicates surprise, at least as far as I understand memes. You all understand it better than I do, probably. But the 4-year-old llama named Winter could hold the key. She was shocked by this. I was just like, oh, of course, llama antibodies. This because llama antibodies have been made as a useful reagent, and they were circulating. They were a tool that X-ray crystallographers used to help proteins crystallize. And this is exactly the paper that they're talking about. This is this is the antibody in blue, I believe. And so they have, this is a, somehow this is able to neutralize, here's the receptor binding domain. It's able to bind right there, and that is big enough to overlap with the actual target of the RBD, the ACE receptor. And so because these clash, if the blue thing is bound, then the red thing won't bind. The other thing about llama antibodies is they are lessons about the Y shape. They don't apply. Llamas make antibodies with a different shape. They are more compact, and therefore they are easier to make. I think you can make them in bacteria recombinantly. But they're compact, and so you can actually make llama antibody to something, and it will crystallize and pull the thing into a crystal with it. So it's one of the ways that crystallographers figured out how to crystallize difficult proteins. And that's why I knew about it before that came along. You can see how it binds, and I just have to show you some amino acids. You have, here's the antibody. You see it has an aspartate binding to an arginine. It just has a really complementary surface, and it's also a compact structure. Here's another picture of it, and there's actually, this is a variation between SARS-CoV-1 and CoV-2. Again, there's not that much variation between the two, and the llama antibody is able to bind both. They actually took this and they measured VF4 binding. This is exactly what you're going to do. It's probably not going to look this clean, but they injected more antibody and they saw more binding. They saw a higher signal. They're able to get equilibrium constant and kinetic constants on and off from that. I just showed this to you because we're going to be doing this. And then the very last thing I want to say is the variation where you bind on the spike is very important. Because if you bind in a place where it can just mutate, it's going to mutate, right? You just put a selective pressure on it, and it's going to respond by avoiding your pressure. But right here, maybe there's a place where the SARS spike can't mutate. What if we had an antibody to that part? Then it couldn't get away from us. So they mapped out the mutability of this spike domain here. And you can see that these exterior regions, like this, is super mutatable. But there's some parts that are more green, and even some parts that are red. They're mostly on the inside. But the parts, if we can find a part, like right here, that's exposed, maybe an antibody to that would be something that SARS can't mutate away from because it can't mutate that residue. Hopefully that's the kind of thinking we want to do. And I just want to say Dr. Gwenwood, the source of our proteins in our project, has done something similar for the adhesion protein, which is like our version of spike. But it's like spike for the MG bacterium. So this is actually MGPC, where we're working with MGPB. But she's colored the points of variation where it varies for patients black. And if you look at that, you look back and forth between these and see where the black is kind of at the place where blue and purple are. It's able to mutate where the black is, and you see that it's sort of on the extreme edges. It even looks like it's almost mutating at the same places. Because the same architecture, the totally different proteins, but they like to mutate in the same places. The exposed places where they can avoid antibodies. So if you get an antibody, most likely you're getting an antibody that binds here, which works great for a while until it mutates and you screw up. We are working not on the top mutatable part, but we're working with the domain down here. And it does not mutate nearly as much. There's hardly any variation in it. So that's one of the reasons why we're looking at it. We're hoping that there's something about this domain. If we find an antibody that works really well, it could have a therapeutic use someday. So that is where I want to end up, because we'll be testing that in the lab.